Welcome, curious minds, to the deep dive. Today, we're embarking on an extraordinary journey, peeling back the layers of plant life to understand how some of the most diverse and vital organisms on our planet build themselves from the ground up. We're diving deep into the angiosperm plant body, structure and development, drawing primarily from raven biology of plants. Think of this as getting the ultimate shortcut, the inside track, really, to understanding how these plants grow. From that tiny, seemingly simple seed into the incredibly complex and beautiful organisms we see all around us. Everything from towering trees to the smallest wildflowers. We're essentially unpacking the blueprint.
Exactly. Our mission today is to trace that fascinating journey. We'll start from the very first cell of a plant embryo, exploring how these fundamental patterns get laid down. Then we'll move through the formation of highly specialized tissues and organs all the way to the mature plant body.
And how they adapt too, right?
Absolutely. We'll also look at how these structures adapt, endure, and thrive in, well, all sorts of different environments. It's about unraveling the intricate biological engineering, the sort of underlying rules that allow a plant to survive, reproduce, and conquer so many ecological niches.
Okay, and to really set the stage for this blueprint, let's consider an intriguing example right from our source material. It paints a great picture. Imagine an endangered Pima pineapple cactus living out in the Sonoran Desert. Soon after it blooms, it produces these sweet green fruits. Desert animals eat them, and in doing so, they actually disperse its large seeds.
A classic dispersal strategy.
Right. But here's a fascinating structural insight. Its sturdy spines. Those aren't just random sharp bits. They're actually modified leaves.
Wow, modified leaves.
Perfectly adapted to provide protection from predators. It's a perfect example of the intricate structural adaptations and developmental innovations we're about to explore, all hidden in plain sight.
That cactus really does highlight the incredible complexity and adaptability we'll uncover. So yeah, our journey will begin microscopically. Inside a seed, we'll examine the earliest cellular divisions that establish the plant's fundamental body plan. Then we'll zoom out a bit to understand the foundational cells and tissues these initial divisions create.
Building blocks, essentially.
Exactly. From there, we'll examine primary growth. That's the growth that extends roots and shoots in length. And finally, we'll delve into the remarkable process of secondary growth. That's what allows woody plants to increase dramatically in girth and stature, building the giants of the plant kingdom.
Okay, let's do it. Section 1. The blueprint of life early development. So let's unpack this incredible journey starting right at the very beginning with Chapter 22, Early Development of the Plant Body. This chapter lays out embryogenesis, the very first phase of seed development. It's where the plant's entire body plan, its fundamental architecture, is precisely laid out.
Exactly. Embryogenesis is just crucial because it establishes two fundamental patterns that will define the entire plant body. First, there's an apical basal pattern.
Apical basal.
Yeah, think of this as the plant's main vertical axis, a clear line running from what will become the shoot tip, way up top, to the root tip at the very bottom. It's the basic up and down direction.
Okay, top to bottom, got it.
Second, there's a radial pattern. This is more like concentric circles of different tissue systems arranged within that main axis.
Like layers in a tree trunk, but right from the start.
Sort of, yeah. It's the inside-out organization, defining layers like the plant's skin, its internal plumbing, and its bulk tissue. This initial setup is foundational for everything that follows.
And the seed itself, which houses this developing embryo, gives the plant a huge advantage, doesn't it? It's way more than just a simple package.
Oh, absolutely. The seed is a truly remarkable evolutionary adaptation. With its mature embryo, its stored food reserves, and that protective seed coat. It dramatically enhances a plant's ability to survive harsh environmental conditions, things like drought or extreme temperatures.
A little survival pod.
Exactly. And it also plays a critical role in facilitating the dispersal of the species, allowing plants to spread to new locations. It's a survival pod and a transportation vehicle all rolled into one.
Let's dive deeper then into the formation of the embryo, the first divisions. So how does this incredible process actually begin? What's the very first step in building a plant?
Well, the initial steps of embryo formation are quite conserved. That means they are remarkably similar across pretty much all angiosperms. It all begins with a single cell, the zygote, which is formed after fertilization. It's nestled right inside the ovule's embryo sac. This one zygote is the blank slate from which the entire plant will develop.
And here's where it gets really interesting for me. The very first division of that zygote is typically asymmetrical and transverse. It's not just a random split down the middle. This immediately establishes the apical basal polarity of the embryo. It's like the plant drawing its first internal map right then and there, designating its top and its bottom.
Yes, that asymmetry is absolutely critical. The division produces two very distinct cells. The upper pole, that's the chalazelle pole, forms a small apical cell. This one is destined to give rise to most of the mature embryo itself, everything from the shoot to the cotyledons, the first leaves.
Okay, so the main body comes from that smaller cell. What about the other one?
The lower pole, the micropylar pole, forms a larger basal cell. This basal cell primarily produces a stalk-like structure known as a suspensor. The suspensor basically anchors the developing embryo within the ovule near the opening called the micropyle.
Polarity sounds a bit technical, but it's just one end being fundamentally different from the other, right? Like a magnet having a north and south pole, or our own bodies having a head and feet. It's like the plant is saying this end is definitely up, that end is definitely down from the very first moment of its existence. It fixes that essential structural axis.
Precisely. This establishment of a structural axis is essential for all higher organisms, really, not just plants. It determines the entire body plan and dictates where all the lateral appendages, like leaves or branches, will eventually be arranged. Without this initial polarity, the plant just wouldn't know how to organize its growth properly. It'd be chaos.
So what happens immediately after that crucial first asymmetrical split? How does it build from two cells?
Well, through a very orderly progression of cell divisions following that initial asymmetric split, the apical cell eventually forms a nearly spherical structure. This is called the embryo proper. And alongside that, the suspensor continues its development. Now, before this spherical stage is fully reached, when it's just a small clump of cells, the developing embryo is referred to as a proembryo.
Proembryo, like a precursor.
Exactly. It's essentially an early stage embryo, still undergoing rapid, mostly undifferentiated cell division.
Okay, moving on to the primary meristems, setting out the tissues. Once we have this proembryo, or the early embryo proper, how does it start to differentiate into different parts? It can't just stay a ball of cells.
Right. Initially, as we mentioned, the embryo proper is just a mass of relatively undifferentiated cells.
Yeah.
But very soon, internal changes begin to establish those concentrically arranged tissue systems we talked about earlier, that radial pattern. This is the first clear expression of the radial pattern, defining the layers from the outside inwards.
And these initial developing tissues, these fundamental building blocks, are called primary meristems, is that right?
Exactly. These are the formative tissues, the precursors to the mature tissues. The outermost cells of the embryo proper undergo specific divisions to form the protoderm.
Protoderm, like first skin.
Pretty much. It will eventually develop into the plant's outer protective layer, the epidermis. Then subsequently, divisions within the embryo proper lead to the formation of the ground meristem. This is the precursor to the ground tissue, essentially the bulk of the plant body, the filling, if you will.
Okay, so outer layer, then the bulk. What's in the middle?
Nestled within that ground meristem, we find the procambium. This is the precursor to the vascular tissues.
The plumbing, xylem and phloem.
Precisely. Xylem and phloem, the plant's internal transport system. And crucially, these three primary meristems, protoderm, ground meristem, and procambium, extend throughout the developing embryo, laying the complete foundation for all future tissues.
Let's talk about embryo development stages. Eudicots versus monocots. The embryo doesn't just, inflate like a balloon. It goes through distinct observable stages. If we were looking under a microscope, what would we see?
You'd see some clear shape changes. That spherical embryo proper is called the globular stage. It's basically just a ball of cells. Then the first leaves, or cotyledons, begin to develop, and this is where we start to see some key differences between the major groups of flowering plants.
Eudicots and monocots, the two big groups.
Right. In eudicots, think beans, sunflowers, roses. These embryos develop two cotyledons. This gives the entire embryo a distinctive heart shape. So, quite fittingly, this stage is known as the heart stage. It really does resemble a tiny green heart under the microscope.
That's a great visual. What about monocots, like grasses or lilies?
In monocots, they form only one cotyledon. As a result, their embryos don't become heart-shaped. They tend to become more cylindrical as they develop.
Okay, so one cotyledon makes it cylindrical, two makes it heart-shaped. And in both types, that apical basal pattern we talked about, the up-down axis, becomes really clear just before these cotyledons emerge, right?
Yes, it becomes very distinct. It effectively partitions the main axis into specific zones. The shoot apical meristem right at the top, then the cotyledon or cotyledons, then the hypocaudal.
That's the bit below the cotyledon.
Yes, the stem-like axis below the cotyledons. Then below that is the embryonic root or radicle, and finally the root apical meristem at the very bottom. It's like a miniature plant, a full body plan laid out.
Wow. And after the heart or cylindrical stage.
As those cotyledons and the embryonic axis continue to elongate, the embryo enters what's called the torpedo stage. It starts to look, well, a bit like a torpedo. During this phase, the primary meristems also extend along the axis. The embryo can either remain straight or it might become curved, depending on the species and space constraints. In monocots, that single cotyledon often becomes surprisingly large, really dominating the entire embryo structure.
Fascinating. Okay, let's move to the suspensor's supporting role and mutational insights. We talked about the suspensor being the anchor for the embryo, but you mentioned it does more than that.
Oh, much more. Unlike in some other plant groups, angiosperm suspensors are highly metabolically active. They're not just passive anchors. They actively support the early embryo by funneling essential nutrients and crucial growth regulators, particularly hormones like gibberellins, towards the developing embryo proper. They essentially feed the embryo.
So like an umbilical cord.
That's a pretty good analogy, yes. It transfers resources. However, the suspensor is relatively short-lived. It undergoes programmed cell death, apoptosis, usually around the torpedo stage of development, so you won't find it in a mature seed. It does its job and then disappears.
Okay, and that leads us to some truly fascinating research involving mutations. You mentioned these give us a window into what might have been development-wise.
Yes, the genetics here are really revealing. What's fascinating is how mutations in certain genes, like one called TUN in the model plant Arabidopsis, reveal the suspensor's hidden potential.
Twin for twin.
Exactly. Normally, the main embryo proper sends inhibitory signals back to the suspensor, basically telling it, stay as a suspensor, don't become an embryo. It suppresses its developmental potential.
Okay.
But in these two mutants, this inhibition pathway is disrupted. And guess what happens? The suspensor cells can actually undergo an embryogenic transformation. They start dividing and developing like an embryo themselves.
No way, so you get twins.
You can get viable twin embryos and sometimes even triplet embryos, all developing from what should have just been the suspensor, all within the same seed coat.
So the main embryo actually tells its potential twin not to develop. That's pretty mind-blowing. Some serious sibling rivalry before birth.
Huh. You could put it that way. It really highlights the intricate cell-to-cell communication that precisely controls plant development. It shows how plastic plant cells can be their way. The suspensor has the potential to be an embryo, but it's actively prevented.
That underscores how critical these signaling pathways are. A delicate balance. Let's discuss the mature embryo and seed features. So, once embryogenesis is complete, after all these stages, what exactly does the mature embryo inside the seed consist of?
Okay, so the fully formed mature embryo of a flowering plant consists of that central axis we discussed, with either one cotyledon if it's a monocot or two cotyledons if it's a eudicot. And critically, at opposite ends of this axis are the apical meristems, one for the shoot and one for the root. These are the pools of stem cells that will drive all future growth after germination.
The growth engines.
Precisely. Now, above the point where the cotyledon attach, there might be an epicaudal. This is a short, stem-like axis that already has some tiny young leaves in its own apical meristem. This whole structure, epicaudal, young leaf, Sam, is called the plumule.
Plumule. That's the embryonic shoot.
That's the embryonic shoot, yes. Below the cotyledons is the hypocaudal. Think hypo, meaning below.
Okay, epicaudal above, hypocaudal below. Makes sense.
And at the lower end of the hypocaudal is the radicle, which is the embryonic root. Sometimes the radicle isn't super distinct, and we just refer to the whole lower part as the hypocaudal root axis.
And the cotyledons themselves, those first seed leaves, they have different jobs depending on the plant species, right? They're not always just leaves.
That's right. Their function varies quite a bit. In many eudicots, like the common garden bean, the cotyledons are really fleshy and packed with stored food. They act as the primary nutrient reserve for the germinating seedling.
So they feed the baby plant.
Exactly. In other eudicots, like the castor bean, or in most monocots, the cotyledons are thinner, more membranous. Their main job isn't storage itself, but rather absorbing stored food from the surrounding tissue called the endosperm.
Ah, the endosperm. That's the nutritive tissue we hear about, like in corn kernels.
Yes, that's a classic example. The endosperm is a separate nutritive tissue formed during fertilization. So in these cases, the cotyledon acts like a bridge or a straw, transferring nutrients from the endosperm to the growing embryo.
In grasses, isn't there a special name for the cotyledon?
There is. In grasses, the single, often massive cotyledon is called the scutellum. It's highly specialized for this absorptive function, with a large surface area pressed against the endosperm.
So the cotyledon can be a storage container or nutrient sponge. Interesting versatility.
Very versatile. And grasses have extra protection too. Both the radical embryonic root and the plumule embryonic shoot in grasses are further protected by sheath-like structures. The coleorhiza covers the radical and the coleoptile covers the plumule. These help them push through the soil during germination.
Extra armor.
Kind of, yes. And finally, wrapping everything up, every seed has a seed coat. This is the protective outer layer developed from the ovule's outer layers, the integuments. It can be thin and papery, like on a bean, or incredibly hard and impermeable to water, like on a coconut. It offers crucial protection against damage, dehydration, and pathogens.
Okay, so that's the mature structure. What about seed maturation? Preparing for the future. What happens after the embryo is fully formed inside?
Right, embryogenesis is just the first phase. The second phase of seed development is maturation. This is a really critical period. This is where the food reserves, whether in the cotyledons or the endosperm, really accumulate. Starch, proteins, oils, depending on the plant. And crucially, the seed undergoes significant dehydration. This process is called desiccation. The water content drops dramatically.
Drying out is key, isn't it, for long-term survival?
Absolutely key. Desiccation is like the plant hitting a pause button. It allows the seed to enter a state of dormancy or quiescence, where its metabolic activity slows down to almost nothing. This allows it to remain viable, to stay alive, but inactive for extended periods, sometimes years, decades, or even, astonishingly, centuries in some cases. And during maturation, the seed coat also hardens, adding to the protection.
So it's fueled up, dried out, and armored up, ready for the wait.
Perfectly put. Ready to wait for the right conditions.
Which brings us to seed germination. Awakening the seed. Once mature, what makes a seed wake up and resume growth? What triggers germination?
Germination isn't automatic. It depends on a precise combination of both external environmental factors and internal physiological states. The three most critical external factors are almost always water, oxygen, and a suitable temperature.
Water seems like the obvious first step for a dry seed.
It is. The process is called imbibition, the rapid uptake of water by the dry seed tissues. This is essential to rehydrate the cells and restart metabolic activity. Water activates enzymes that were already present in the seed, and it triggers the synthesis of new enzymes needed to digest and mobilize those stored food reserves we talked about.
And oxygen.
Oxygen is crucial for efficient energy production through aerobic respiration. Initially, when the seed is first taking up water, some glucose breakdown might start anaerobically without oxygen. But the aerobic pathway, which yields much more energy, quickly takes over once the seed coat ruptures or becomes permeable enough to allow oxygen in.
So that's why waterlogged soil can be bad for germination. Too much water blocks the oxygen.
Exactly. It prevents aerobic respiration, and the seedling essentially suffocates. And temperature is also critical. Most seeds have a specific temperature range within which they will germinate, often with an optimum somewhere between 25 degrees to 30 degrees C, typical room temperature, or slightly warmer.
Some seeds also need light, right? Like lettuce.
Yes, particularly small seeds, like lettuce or many weeds, often require exposure to light as a germination cue. This often ensures they only germinate when they are near the soil surface, not buried too deep.
Okay, water, oxygen, temperature, sometimes light. But this raises an important question, leading us to seed dormancy, a survival strategy. Why doesn't every seed just germinate as soon as conditions seem right? Why the delay in some cases?
That's where dormancy comes in, and it's a really fascinating and powerful survival mechanism. Unlike seeds that are merely quiescent, meaning they're just waiting for favorable conditions and will germinate readily once they get them truly dormant, seeds will not germinate even if external conditions like water, oxygen, and temperature appear to be perfectly ideal.
So dormancy is like an extra lock on the germination process.
Exactly. It's an internal block. It ensures that the seedling emerges only when its chances of long-term survival are truly highest, not just during a brief, favorable spell. Think about a warm snap in autumn. A quiescent seed might germinate, only to be killed by the first frost. A dormant seed wouldn't be fooled.
Makes sense. What causes this dormancy? Is it the coat or the embryo itself?
It can be either, or sometimes both. We talk about coat-imposed dormancy and embryo dormancy. Coat-imposed dormancy can result from several things. The seed coat might be physically impermeable to water or oxygen, stopping germination before it starts. Or it might be mechanically rigid, literally preventing the embryo from physically breaking out. Or sometimes the coat contains chemical growth inhibitors that need to be leached out or broken down.
Like a chemical lock.
Yes. Then there's embryo dormancy. This often involves the internal balance of plant hormones within the embryo itself, specifically abscisic acid, ABA, which generally promotes dormancy, and gibberellic acid, GA, which tends to promote germination. The ratio between these two is often key.
Hormonal control.
Right. Also, sometimes the embryo itself is physiologically immature, even when the seed is shed from the parent plant. It needs to undergo a period of after-ripening. This involves a series of enzymatic and biochemical changes within the embryo, often triggered by specific environmental cues. For instance, many temperate plants require a period of prolonged cold exposure, like overwintering, to break dormancy. This ensures they only germinate in the spring when conditions are truly favorable for seedling growth.
So dormancy acts as a sophisticated internal clock or calendar, ensuring the plant isn't fooled by, say, a warm day in winter and sprouts too early only to be killed by a later freeze. It's a critical evolutionary advantage for survival.
Exactly. It's of immense survival value. And dormancy can be complex. There's primary dormancy, which is established during seed maturation on the parent plant. But seeds can also enter secondary dormancy if they experience unfavorable conditions after primary dormancy has already been broken.
Wow, so they can go back to sleep.
In a way, yes. It adds another layer of caution. And there are other fascinating dormancy-breaking triggers adapted to specific environments. Some seeds need to pass through the digestive tract of an animal. The acid or grinding action breaks down the coat. Others in deserts might need heavy rainfall to leach out inhibitors. Some need mechanical cracking or even the intense heat of a fire to trigger germination. Plant germination strategies are incredibly diverse and finely tuned to their specific ecological niches.
Really tailored survival plans. Okay, so let's assume dormancy is broken, conditions are right. From embryo to adult plant, the seedling emerges. Once germination finally kicks off, what's the first thing we usually see pushing out of that seed coat?
The root is almost always the first structure to emerge. Think about it makes perfect biological sense.
Anchorage and water first.
Precisely. It needs to immediately anchor the fragile seedling in the soil and, crucially, start absorbing vital water and minerals to fuel further growth. This initial root that emerges from the radicle is called the primary root, or often the taproot. It typically grows straight down and then develops lateral roots branching off it.
So that's why gardeners are always so careful with the root ball when transplanting seedlings. That primary root is key.
Very key, especially in plants with taproot systems. However, in monocots, as we mentioned before, this primary root is often short-lived. The main mature root system develops instead from stem-borne adventitious roots. These arise from the base of the stem, forming a much shallower but very dense and tenacious fibrous root system, like you see in grasses.
Okay, the root gets established. Then how does the shoot, the part that needs sunlight for photosynthesis, emerge safely from the soil? It must be quite delicate.
It is delicate, and plants have evolved clever ways to protect that growing shoot tip as it pushes through abrasive soil. There are two main strategies for shoot emergence. The first is epigeous germination, epi meaning above. Here, the hypocotyl, remember, the stem part below the cotyledons elongates rapidly. It forms a hook shape.
A hook?
Yes, an archer hook. This hypocotyl hook pushes up through the soil first, dragging the delicate cotyledons and the plumule, the embryonic shoot tip behind it, protecting them. Once it breaks the surface, the hook straightens out, pulling the cotyledons and shoot tip into the air. Think of garden beans, castor beans, or even onions. Their cotyledons come above ground. Sometimes these cotyledons even turn green and photosynthesize for a while.
Okay, so the hypocotyl hook pulls everything up. What's the other way?
The second is hypogeous germination. Hypo meaning below. In this case, the cotyledons stay below ground. Instead of the hypocotyl, it's the epicotyl, the stem part above the cotyledons, that elongates and forms the protective hook. This epicotyl hook lifts only the plumule, the shoot tip with its young leaves, above the ground. The cotyledons remain buried in the soil, usually just transferring their stored food reserves until they're depleted, and then they decompose. Peas and maize, corn, are classic examples of hypogeous germination.
So either way, The plant protects that super important shoot tip as it pushes through the soil, either by pulling it up from below or lifting it up from above, always using a hook for protection.
Yes, it's a very effective protective mechanism. Once that shoot is safely established above ground, the activity of its apical meristem takes over, leading to an orderly sequence of new leaves, nodes, where leaves attach, and internodes, the stem segments between nodes. And axillary buds develop in the leaf axils, ready to grow into lateral shoots or branches.
But getting to that point must be tough.
It is. This initial period of seedling establishment, from germination until the young plant has produced enough leaves to be self-sufficient through photosynthesis, is arguably the most crucial and vulnerable phase in a plant's entire life cycle. Many don't make it past this stage.
A perilous beginning. Okay, let's move on. Section 2. Building blocks, cells, and tissues. So Chapter 22 showed us that incredible journey from a single fertilized cell to a tiny embryo, packed in a seed, ready to sprout. But as you said, that's just the very beginning. Now let's zoom in with Chapter 23, cells and tissues of the plant body. We need to understand how this embryo actually becomes a complex, multicellular plant. And that involves the continuous activity of meristems and the formation of all the specialized tissues.
That's exactly right. Most of the plant development, we actually see the increase in size, the formation of complex structures happens after embryogenesis, and this ongoing growth is all thanks to these amazing regions called meristems.
The growth zones we mentioned.
Yes, regions of actively dividing cells. What's special about meristem cells is that they retain their potential for division long into the plant's life. They're essentially like perpetually young stem cells for the plant. We've already met the apical meristems, found at the very tips of all roots and stems. They are primarily responsible for the plant's primary growth. That means the extension of the plant body in length, making roots longer, making stems taller, and the formation of all its primary tissues.
So they're literally the engines of elongation, constantly adding new material at the ends.
Essentially, yes. Within these meristems, there are special cells called initials. When an initial divides, one daughter cell remains an initial. It stays in the meristem, maintaining that capacity for future growth. The other daughter cell becomes a derivative.
A derivative, what does that do?
The derivative cell might divide a few more times, but eventually it will undergo differentiation. That means it will mature and specialize into a specific type of cell, becoming part of a particular tissue. And remember, those primary meristems, the protoderm, procambium, and ground meristem that were first established back in the embryo.
Yes, the precursors for the main tissue systems.
Right. Those primary meristems are continuously extended by the activity of the apical meristems. As the derivatives they produce move away from the apex, they differentiate into the plant's mature tissue systems. The epidermis from protoderm, the primary vascular tissues, xylem and phloem from procambium, and the ground tissues, like cortex and pith, from the ground meristem.
Okay, now diving into growth, morphogenesis, and differentiation, what makes cells different? Plants don't just get bigger, they grow into specific shapes. That's morphogenesis. And their cells become highly specialized for different jobs. It's differentiation. But how does a plant cell know what it's supposed to become? This seems like a central question.
It absolutely is a profound question in plant biology and an area where research has yielded some really insightful findings. Now, while cellular differentiation certainly depends on controlling gene expression, turning specific genes on or off, so different cells make different proteins, what's truly striking about plants, and quite different from animals in many ways, is that the ultimate fate of a developing plant cell, what kind of specialized cell it will eventually become, seems to be determined primarily by its final position within the developing organ.
Its position, not its ancestry.
Largely, yes. It's less about its lineage or who its parent cells were, and more about where it ends up in the grand scheme of the developing tissue or organ.
So it's not about who your parents were, but where you end up living in the plant body. That's really different.
It is different. Studies, including some very elegant experiments involving laser ablation to remove specific cells and roots, have demonstrated this principle clearly. If an undifferentiated cell is somehow displaced, or if its neighbors are removed, it will often differentiate into a cell type appropriate for that new location, overriding its original expected fate based on lineage.
Wow. So they're constantly sensing their surroundings and adjusting their identity.
It strongly indicates that plant cells are actively communicating positional information to one another. They're essentially telling their neighbors, you are in this layer, or you are next to a vascular strand, and that information guides the differentiation process. It's like a cellular GPS system guiding development.
That's a real aha moment. It suggests a fascinating flexibility and continuous communication network within the plant, allowing it to adapt its development. Incredible.
It truly is. It points to a level of developmental plasticity that's quite remarkable.
Okay, so these cells differentiate based on position. Now let's look at the three tissue systems organizing the plant body. How do these differentiated cells organize themselves into the larger structures of the plant?
Right. These differentiated cells don't just exist randomly. They group together to form larger structural and functional units called tissues, things like parenchyma tissue or xylem tissue. And these different tissues are, in turn, grouped into three fundamental continuous tissue systems that essentially run throughout the entire plant body, from root tip to shoot tip. They form the basic organizational framework.
Three systems, what are they?
They are. One, the dermal tissue system. This is the plant's outer protective covering effectively its skin, usually the epidermis or later, the periderm. Two, the vascular tissue system. This functions as the plant's internal transport or circulatory system, moving water, minerals, and sugars. That's the xylem and phloem. Three, the ground tissue system. This makes up the bulk of the plant body, essentially everything else, the body mass filling the space between the dermal and vascular tissues.
Okay, dermal, vascular, ground. And these three systems directly correspond to those three primary meristems we discussed earlier, right? Protoderm makes dermal, procambia makes vascular, and ground meristem makes ground tissue.
Exactly, there's a direct lineage. And the basic arrangement is almost always a radial pattern. The vascular tissues are typically embedded within the ground tissue, and the dermal tissue forms the outer covering.
So the differences we see between roots, stems, and leaves are mostly about how these systems are arranged relative to each other.
Precisely. The differences largely come down to how the vascular tissues and the ground tissues are distributed within the organ. For instance, in a typical eudicot stem, the vascular strands might form a cylinder arranged around a central ground tissue region called the pith. But in a root, the vascular tissues might form a solid central cylinder with the ground tissue, cortex, only surrounding it. It's the spatial organization that differs.
Okay, and tissues themselves can be simple or complex.
Right. Simple tissues are composed of only one type of cell, like parenchyma tissue. Complex tissues, like xylem or phloem, are made up of two or more different cell types working together.
Let's dig into each system, starting with the ground tissues, the bulk of the plant. This makes up most of the plant, you said.
Yes, the ground tissue system is typically the most extensive. It consists of three main types of simple tissues. Let's start with parenchyma tissue.
This sounds like the most common one, the general filler tissue.
It is in many ways. Parenchyma cells are the most numerous cell type in many plants. They are typically living cells at maturity, and they are incredibly versatile. You find them forming continuous masses throughout the plant body, making up the cortex and pith of stems and roots, the mesophyll tissue inside leaves where photosynthesis happens, and even the soft, fleshy parts of fruits.
What are their main jobs? These sound like jacks of all trades.
They really are. They're involved in major metabolic processes like photosynthesis, respiration, and secretion. They also function extensively in the storage of food reserves like starch, proteins, or oils. They conduct substances over short distances, and remarkably, many parenchyma cells retain their capacity for cell division even when mature. This makes them key players in wound healing and regeneration. They can divide to repair damage or even form new roots or shoots in some cases.
Very versatile indeed. Okay, what's the next ground tissue? Collenchyma tissue.
Collenchyma cells are also living cells at maturity. They are typically elongated, and their defining feature is their unique cell wall. They have primary walls that are unevenly thickened, particularly at the corners. Crucially, these thickened walls are not lignified. They don't have the rigid lignin polymer found in wood. This means the walls remain soft and pliable. In fresh tissue, they often have a glistening appearance.
Like the strings in a celery stalk.
Yeah.
Are those collenchyma?
That's a perfect example. Those tough but flexible strings are almost entirely collenchyma tissue.
So their main job is flexible support, especially in young growing parts.
Exactly. Because they are living cells and their walls are pliable, they can actually continue to develop and thicken their walls as an organ is elongating. This makes them perfectly adapted for providing mechanical support to young, growing stems, leaf stalks, petioles, and along the veins and leaves without restricting that growth. You find them typically just beneath the epidermis in stems and petioles.
Okay, flexible support for growing parts. Now what about really strong, rigid support, especially once growth has stopped?
For that, the plant relies on sclerenchyma tissues. These cells provide rigidity and strength. The key difference is their cell walls. Sclerenchyma cells develop very thick and usually heavily lignified secondary walls. Lignin is that hard, rigid polymer that makes wood strong. And importantly, sclerenchyma cells often lack protoplasts, meaning they are dead at maturity. Their function relies entirely on their strong, persistent cell walls.
Dead but strong.
Dead but strong. They are critical strengthening elements in plant parts that are no longer elongating.
And they come in two main forms, right? Fibers and sclerids.
Yes, two main types. Fibers are generally long, slender cells that usually occur in strands or bundles. Think of the economically important bast fibers used to make ropes and textiles, like from hemp, flax, or jute. Those are sclerenchyma fibers. Sclerids, on the other hand, are much more variable in shape. They can be relatively short and cuboidal, or branched, or star-shaped. They often occur singly or in small aggregates rather than large bundles.
Where would we find sclerids?
They contribute to the hardness of things like seed coats and nutshells. The hard stone or pit of fruits like peaches, cherries, plums, and olives is made primarily of sclerids, and that slightly gritty texture you sometimes get when eating a pear.
Yeah, what is that?
Those are clusters of sclerids, sometimes called stone cells, scattered in the soft parenchyma tissue of the pear flesh.
Fascinating. Okay, that covers the ground tissues. Let's move to the vascular tissues. The plant's transport system. This system is absolutely vital, linking all parts of the plant.
It is the plant's lifeline, no doubt. It's responsible for the long-distance transport of water, dissolved minerals, and the sugars produced during photosynthesis. It comprises two complex tissues, xylem and phloem, which together form a continuous network extending from the deepest roots all the way to the tips of the highest leaves.
Let's start with xylem, water and minerals. So xylem is the principal water conductor, moving water up from the roots.
Correct. Its primary function is the bulk transport of water, but it also transports dissolved minerals absorbed from the soil. Additionally, the thick, lignified walls of xylem cells provide significant structural support to the plant body, and xylem parenchyma cells also function in food storage. Xylem can be primary, formed during primary growth from procambium, or secondary, formed during secondary growth from the vascular cambium.
And the main conducting cells are called trachary elements. You mentioned two types before. Tracheids and vessel elements. What's the key difference again?
Both are elongated cells. Both have thick, lignified secondary walls. Both lack protoplasts, they are dead at maturity, forming hollow tubes. And both have pits, which are thinner areas in the secondary wall where water can move laterally between adjacent cells. The key difference lies in vessel elements. Vessel elements have perforations, which are areas completely lacking both primary and secondary walls. These perforations are usually located on the end walls where vessel elements connect.
So like actual holes at the end.
Exactly, like holes, or even the complete removal of the end wall. This allows vessel elements to join end to end to form long, continuous, multicellular tubes called vessels. These vessels act like efficient pipelines, allowing for relatively unobstructed bulk flow of water. Vessels are considered the principal water-conducting structures in most angiosperms.
Like plumbing pipes, essentially. Very efficient.
A very good analogy. Tracheids, however, lack these perforations. Water has to move between adjacent tracheids primarily through pit pairs. This makes them generally less efficient for bulk water transport than vessels. Tracheids are the sole water-conducting cells in most seedless vascular plants like ferns and gymnosperms like conifers, though many angiosperms actually possess both vessels and tracheids in their xylem.
And how do these trachary elements adapt during growth? You mentioned stretching.
Right. In the parts of the plant that are still elongating, the first formed primary xylem, called protoxylem, needs to be stretchable. So its secondary walls are deposited in patterns that allow for this, typically as rings annular or spirals, helical. This reinforces the cell, but still allows it to stretch like a spring as the organ grows longer.
Clever design.
Very clever. Later in the metaxylem, primary xylem formed after elongation stops, and in all secondary xylem, the trachary elements, often called pitted elements, become much more rigid. Their secondary walls cover almost all of the primary wall surface except at the pits.
And the actual formation of these elements involves that pretty dramatic process you mentioned earlier, programmed cell death.
Indeed. The differentiation of a trachary element is a classic, clear example of programmed cell death or apoptosis in plants. The living part of the cell, the protoplast, is completely dismantled and removed in a highly controlled way. All that's left behind are the cell walls, forming that non-living, unobstructed conduit perfect for water transport. Xylem isn't just conducting cells, though. It also contains living parenchyma cells for storage and often sclerenchyma fibers for additional support.
Okay, so that's water and minerals sorted by the xylem. Now for phloem, food transport. If xylem moves water mostly up, phloem is the system that moves food, primarily sugars, all around the plant wherever it's needed.
Yes, that's its primary role. Phloem conducts the sugars, or photosynthates, produced during photosynthesis, usually in the leaves, to other parts of the plant where they are needed for growth, like root tips, shoot tips, developing fruits. Or for storage, like in roots, tubers, or seeds. Like xylem, phloem can be primary, from procambium, or secondary, from vascular cambium.
And its conducting cells, they're different from xylems, right? They stay alive.
They do stay alive, which is a key difference. The main conducting cells in the phloem of angiosperms are called sieve tube elements. They're quite unique because they remain alive at maturity, but they undergo a very selective partial breakdown of their protoplast. They lose their nucleus, their vacuole membrane, tonoplast, ribosomes, and Golgi apparatus. But critically, they retain their plasma membrane, smooth endoplasmic reticulum, plastids, and mitochondria. This selective breakdown essentially clears out the cell's interior, allowing it to function as a more open conduit for transport, yet it remains a living, metabolically active cell.
That's a crucial difference from xylem, where the cells die completely to become functional pipes. This allows for active transport in phloem, presumably.
It does. The transport mechanism in phloem is different and relies on living cells. A key feature in angiosperm sieve tube elements are the sieve areas on their walls, particularly concentrated on the end walls where they connect, forming sieve plates. These are areas with large pores lined with plasma membrane, allowing connection between adjacent sieve tube elements. Another interesting component is P protein, phloem protein. It forms distinct bodies in young sieve tube elements and then disperses throughout the cell lumen in mature ones. What's its function? Upon wounding, say, if an aphid pierces the phloem, P protein rapidly accumulates at the sieve plates, forming slime plugs.
Slime plugs, like a quick clot.
Exactly like a quick clot. It prevents the pressurized contents of the phloem, the valuable sugary sap, from leaking out. It's a rapid wound sealing mechanism. Some legumes even have specialized crystalline P protein bodies called forasomes that can rapidly and reversibly occlude the sieve pores in response to stimuli.
Wow. And these sieve tube elements don't work alone, do they?
No, they are intimately associated, both structurally and functionally, with companion cells. Companion cells are specialized parenchyma cells that arise from the same mother cell as the sieve tube element they are associated with. Unlike the sieve tube element, the companion cell retains a full complement of organelles, including a prominent nucleus and dense cytoplasm with lots of ribosomes and mitochondria. Through numerous plasmodesmata, cytoplasmic connections, linking the two cells, the companion cell effectively controls the metabolism and activity of the enucleated sieve tube element. It loads sugars into the sieve tube and keeps it alive and functioning.
So the companion cell is like the life support system and control center for the sieve tube element.
That's an excellent way to put it. Phloem tissue also typically contains parenchyma cells for storage, and sometimes fibers and sclerides for additional support, similar to xylem.
Okay, that covers the vascular system. Lastly, let's move to the dermal tissues, the plant's outer protection. These are the layers that form the interface between the plant and its environment.
Precisely. These are the protective layers, crucial for mediating the plant's interaction with the outside world, preventing water loss, protecting against physical damage and pathogens, and regulating gas exchange.
The main one is the epidermis. This is the outermost layer of the primary plant body, right? The skin.
It is. The epidermis is typically a single layer of tightly packed, often flattened cells. Its primary role is mechanical protection. On the aerial parts of the plant, stems, leaves, flowers, the outer surface of the epidermal cells is covered by a cuticle. The cuticle is composed mainly of cutin, a waxy, fatty substance, often embedded with or covered by additional waxes. This cuticle is very effective at minimizing water loss through evaporation from the plant surface, which is crucial for land plants. These surface waxes can sometimes form a visible whitish or bluish layer, often called a bloom, on leaves or fruits like grapes or plums.
And embedded within this epidermis are the gateways for gas exchange, the stomata.
Yes, stomata, singular stoma, are essential. They are basically small pores in the epidermis, each regulated by a pair of specialized, often kidney bean-shaped guard cells. These guard cells contain chloroplasts and can change shape to open or close the pore.
What do they control?
They control the exchange of gases between the plant's internal tissues and the atmosphere. Primarily, this means taking in carbon dioxide needed for photosynthesis and releasing oxygen as a byproduct. But crucially, they also regulate the loss of water vapor from the plant in a process called transpiration. They are most abundant on leaves, often concentrated on the underside.
Many plants also have fascinating hairs or other outgrowths from the epidermis called trichomes. They're not just fuzz, are they?
Definitely not just fuzz. Trichomes are incredibly diverse in their structure and function. They can be single-celled or multicellular, branched or unbranched, glandular or non-glandular. Their functions are just as varied. In epiphytic air plants, specialized trichomes can absorb water and minerals directly from the atmosphere. In plants like the saltbush, they secrete excess salt to prevent toxic accumulation in the tissues. Many trichomes provide physical defense against insects, making it hard for them to walk or feed, or they might reflect excess sunlight.
And glandular ones.
Glandular hairs can produce a wide range of substances, including sticky insect traps, volatile oils that act as chemical defenses or attractants or irritants. The brand non-glandular trichomes of the model plant Arabidopsis have been a fantastic system for studying cell fate determination. Genes like GLBR1, GL1, are needed for a cell to even initiate becoming a trichome, while others, like transparent testaglabra, TTG, seem to be involved in inhibiting neighboring epidermal cells from also becoming trichomes, ensuring proper spacing. It again shows how precise cell-to-cell communication controls pattern formation.
So much complexity in just a hair. Okay, so the epidermis covers the primary plant body. But what happens when stems and roots get thicker with secondary growth, like in a woody tree? That single layer of epidermis can't possibly stretch enough.
You're absolutely right, it can't. It gets stretched, torn, and eventually sloughed off. That's when the epidermis is replaced by a secondary protective tissue called the periderm. The periderm essentially becomes the new functional outer skin of woody stems and roots.
And the periderm itself is complex, right? Not just one layer.
Correct. The periderm actually consists of three distinct parts. One, the cork cambium, also called the phellogen. This is the meristematic layer, usually arising from cortical cells just beneath the original epidermis that produces the other two layers. Two, cork, also called phellum. This is produced to the outside of the cork cambium. Cork cells become heavily impregnated with suberin, another waxy, waterproof substance, and often lignin as well. They are typically dead at maturity and form the tough, protective outer layer of the bark. Commercial cork from the cork oak tree is just a massively developed layer of phellum. 3. Phelloderm. This is produced to the inside of the cork cambium. It's usually only one or a few layers thick and consists of living, parenchyma-like cells that resemble the cortical cells from which the cork cambium originated.
Cork cambium, cork, phelloderm. Together they make the periderm. And I've noticed those small, raised, often rough-looking spots on the trunks and branches of trees.
Yeah.
Are those related to the periderm?
Yes, those are lenticels. Remember how the cork layer is heavily suberized and impermeable to gases?
Right, seals it off.
Well, the living tissues inside the stem or root still need to respire. They need oxygen. Lenticels are specialized regions within the periderm where the cork cambium produces cells that are much more loosely arranged with lots of intercellular air spaces. These spongy areas provide pathways for vital gas exchange, oxygen in, carbon dioxide out through the otherwise impermeable bark, connecting the internal tissues to the atmosphere. They often form underneath the original stomata of the epidermis and continue to develop and function as the plant ages and the bark thickens.
Clever way to allow breathing through the armor. Okay, that wraps up the basic cells and tissues. Section 3, root systems, anchorage, absorption, and adaptation. We've built up the fundamental cells and tissues. Now let's transition to Chapter 24, the root, structure and development. It's time to explore how these building blocks come together to form one of the essential yet often hidden organs of the plant, the roots, delving into that world beneath our feet.
The root truly is the unsung hero of the plant world. It performs two primary and absolutely vital functions: anchorage, firmly securing the plant in the soil against wind and gravity, and absorption, taking up essential water and dissolved mineral nutrients from the soil. But their roles don't stop there. Roots also often serve as major sites for storage of food reserves, particularly carbohydrates like starch. They are involved in conduction, transporting water and minerals upwards and sugars downwards. And interestingly, roots also synthesize important plant hormones, like cytokinins and gibberellins, which are then transported to the shoot where they influence growth and development.
A multitasking organ hidden away. We mentioned primary roots and different root systems way back in the embryo stage. Let's elaborate now on root system types and architecture in the mature plant. How do they typically look?
Certainly. In many seed plants, particularly eudicots and gymnosperms, the primary root that emerges from the embryo continues to grow downwards, becoming the dominant root of the system. This is called a taproot. Lateral roots then branch off this main taproot, forming a taproot system. These systems can penetrate incredibly deep into the soil, seeking out water far below the surface. We mentioned that record from Boccia albatrunca in the Kalahari Desert Roots, reaching 68 meters down.
That's over 220 feet.
Yeah.
Still boggles my mind. Just imagine that hidden network anchoring and feeding everything we see above ground.
It's truly astounding. In monocots, however, the situation is usually different. The primary root that emerges from the seed is often short-lived. It doesn't persist or become dominant. Instead, the main functional root system arises from numerous adventitious roots. Remember, adventitious means developing from an unusual place in this case. They usually arise from the base of the stem, near the soil surface.
Ah, so not from the embryonic root.
Correct. These numerous stem-borne roots branch further, forming a dense mesh-like fibrous root system. This type of system is common in grasses and other monocots. It's generally shallower than a taproot system, but very tenacious and excellent at holding soil together, which is why grasses are so good at preventing soil erosion.
So taproots for depth, fibrous roots for surface area and holding soil. Plants adapt their root systems to their needs and environment.
Absolutely. Roots are incredibly sensitive to environmental cues. They respond to gravity, moisture gradients, temperature differences, and patches of high nutrient concentration in the soil. They exhibit remarkable developmental plasticity. For example, if a root encounters a pocket rich in nitrates or phosphates, it will often rapidly proliferate lateral roots specifically within that zone to maximize uptake.
They actively forage for resources.
They actively forage. And a critical aspect of overall plant growth is maintaining a functional balance between the root system and the shoot system. This is often expressed as the root-shoot ratio. Essentially, the plant needs to maintain an equilibrium between its photosynthesizing leaf area, the shoot, and its water and mineral-absorbing root surface area. If you damage one system, say, prune the roots heavily during transplanting the growth of the other system, the shoot, will often be temporarily slowed until that balance can be reestablished.
That's why gardeners sometimes cut back the tops of plants when they transplant them, to compensate for root loss.
Precisely. Cutting back the shoot reduces the demand for water and minerals, giving the damaged root system a chance to recover and reestablish that crucial balance.
Okay, let's explore the root tip, a growing and sensing machine. Let's trace the growth right down to the very end of the root. It's covered by a special cap, isn't it? Protecting the delicate growing point.
Yes, exactly. The very apex of the root is covered by a structure called the root cap. It's like a little thimble or helmet made of living parenchyma cells. Its primary job is indeed protection. It shields the delicate apical meristem located just behind it as the root tip pushes its way through the often abrasive soil particles.
A protective helmet. Does it do anything else?
It does. It's quite dynamic. The root cap cells, especially the outer ones, secrete large amounts of mucilage. This is a highly hydrated polysaccharide slime.
Slime.
Yes, essentially a slime layer. It acts as a lubricant, reducing friction and making it easier for the root to penetrate the soil. It also helps maintain contact between the root and soil particles, which is important for water absorption.
So a protective helmet and a slippery coating.
Yeah, and there's more. The root cap is increasingly seen as a multifunctional molecular relay station. Its peripheral cells, sometimes called border cells, are programmed to detach from the root cap as it moves through the soil. These detached, living cells might play roles in protecting the root tip from pathogens, maintaining root-soil contact, helping to mobilize nutrients in the immediate vicinity, and maybe even acting like tiny ball bearings to further reduce friction.
Wow, shedding cells to help it move. That's wild. And it senses things, too.
Yes. The central column of cells within the root cap, called the columella, contains specialized plastids called ameloplasts that act as stalithes gravity sensors. This allows the root cap to perceive gravity, gravitropism, and guide the root's growth downwards. It's also involved in sensing water potential gradients, hydrotropism, helping the root grow towards sources of moisture.
A very smart helmet. How is the apical meristem itself organized, just behind the cap?
Its organization can vary. In what's called the closed-type organization, like you see in maize, the root cap, the vascular cylinder, and the cortex each originate from their own independent layers of initial cells in the meristem. In the open type, like an onion, all these regions seem to trace back to one common group of initials. It's less clearly compartmentalized.
And there's also that peculiar quiet zone we mentioned earlier, right in the middle of all this active cell division.
Yes, the quiescent center. This is a relatively inactive region, usually located just behind the root cap within the core of the apical meristem, specifically the promeristem, which includes the initials and their immediate derivatives. Cells here divide much less frequently than the surrounding meristematic cells. While its exact roles are still debated, it seems crucial for maintaining the overall organization and long-term integrity of the root meristem. It might act as a reservoir of undamaged stem cells and can even repopulate or regenerate parts of the meristem if they get injured.
A reserve command center. So the root isn't just growing evenly along its whole length from the tip backwards.
No, definitely not. We can functionally divide the root tip region, just behind the root cap and apical meristem, into distinct zones based on cellular activity. First is the region of cell division, which is the apical meristem itself plus the promeristem, where new cells are being produced. Immediately behind that is the region of elongation. Here, the cells produced by the meristem stop dividing but rapidly enlarge, primarily by taking up water into their vacuoles. This cell elongation is what actually drives most of the increase in the root's length, pushing the tip forward through the soil.
So division makes new cells, elongation pushes the tip. What's next?
Behind the region of elongation is the region of maturation or differentiation. This is where the cells, having reached their final size, differentiate and mature into their specialized types, forming the primary tissues of the root, like the epidermis, cortex, and vascular cylinder. And importantly, this is also the region where root hairs are produced.
Why there? Why not closer to the tip?
Because root hairs are delicate extensions of epidermal cells. If they formed in the region of elongation where the root surface is actively moving past soil particles, they would be immediately sheared off and destroyed. They only form once that part of the root has stopped elongating and is stationary relative to the soil, allowing them to penetrate between soil particles and absorb water and nutrients effectively.
Timing is everything. That makes perfect sense.
And the timing of tissue maturation within this region is also interesting. The first formed primary phloem elements, protophloem, which transport sugars down to fuel the growing tip, mature closer to the root tip than the first formed primary xylem elements, protoxylem. This indicates the immediate and critical need for food transport to sustain that energy-intensive root growth and cell division at the apex.
Fascinating sequence. Okay, once we're beyond the actively growing tip in the mature region, let's look at the primary structure of the root. Layers of function. What does the root's internal structure look like in cross-section?
In its primary state, before any secondary growth, the root's internal structure is generally simpler than that of a stem. It lacks nodes and internodes, for instance. If we take a cross-section through the region of maturation, we typically see three distinct concentrically arranged tissue systems: the outermost epidermis, the large cortex beneath it, and the central vascular cylinder, also called the stele.
Let's start with the epidermis, the absorbing surface.
In young, actively absorbing roots, the epidermis consists of thin-walled, closely packed cells. Crucially, unlike the stem epidermis, the root epidermis typically lacks a well-developed cuticle, or has a very thin one. This facilitates the absorption of water and minerals directly from the soil. And as we just discussed, the efficiency of this absorption is vastly increased by the presence of root hairs. Remember, these are not multicellular appendages. They are tubular, single-cell extensions of individual epidermal cells. They dramatically increase the surface area in contact with the soil.
You give that amazing statistic for rye.
Yes, a single 4-month-old rye plant was estimated to have something like 14 billion root hairs, providing a total absorbing surface area of about 401 square meters. That's roughly the size of 2 tennis courts, just from the root hairs of one plant. It shows how vital they are.
Incredible surface area just for absorption. Okay, beneath the epidermis is the cortex, storage and protection.
Correct. The cortex usually makes up the largest part of the volume of a young root in cross-section. It's primarily composed of parenchyma cells, which commonly store starch as food reserves. These cortical cells often lack chlorophyll as they are underground and not exposed to light. There are typically prominent intercellular spaces between the cortical cells, which are vital for aeration, allowing oxygen to diffuse from the soil to the living cells of the root. The innermost layer of the cortex, bordering the central vascular cylinder, is a very distinct and functionally critical layer called the endodermis.
The endodermis, this has those special Casparian strips. What exactly are those and why are they so important? You mentioned a checkpoint earlier.
Yes, the Casparian strip is absolutely key to understanding root function. It's not just a simple wall thickening. It's an integral band-like portion of the primary cell wall of every single endodermal cell. This band runs along the radial walls, the side walls connecting adjacent endodermal cells in the ring, and the transverse walls, the top and bottom walls. This band is impregnated with water-repellent substances, primarily suberin and sometimes lignin as well. Imagine it like a waterproof gasket or sealant completely encircling each cell within the endodermal layer.
A waterproof seal between cells, what does that do?
Its crucial function is to block the apoplastic pathway. That's the pathway for water and dissolved solutes moving between cells through the interconnected cell walls and intercellular spaces of the cortex. Because the Casparian strip is impermeable, water and solutes traveling via this apoplastic route hit a dead end when they reach the endodermis.
So it forces everything that wants to get into the vascular system to actually go through the living endodermal cells.
Exactly. It forces transport to switch to the symplastic pathway movement through the cytoplasm of the cells, crossing the plasma membrane. All substances entering or leaving the central vascular cylinder must pass through the selectively permeable plasma membrane and the cytoplasm of the endodermal cells. This gives the plant precise control over what ions and molecules enter its vascular system, its internal plumbing. It acts as a selective filter, preventing unwanted or toxic substances from reaching the rest of the plant. It's the plant's ultimate quality control checkpoint for water and minerals absorbed from the soil.
That's a brilliant life or death filtration mechanism hidden underground. Does the endodermis change as the root gets older?
It often does. In older roots, especially those undergoing secondary growth, the endodermal cells frequently deposit additional layers of suberin lamellae and sometimes thick layers of cellulose over their entire inner surface, making them even more impermeable. However, even in these heavily fortified endodermal layers, there are often some cells that remain relatively thin-walled for a longer time. These are called passage cells, and they are typically located directly opposite the protoxylem poles of the vascular cylinder, perhaps facilitating transfer between the xylem and the cortex.
Okay, so inside the endodermis is the vascular cylinder, steel, the core of transport.
Yes, this is the central core of the root containing the primary vascular tissues, the xylem and phloem, responsible for long-distance transport. Surrounding these vascular tissues, just inside the endodermis, is the pericycle. The pericycle is typically one or more layers of non-vascular cells, usually parenchyma, that originate from the procambium. The pericycle is remarkably versatile and retains meristematic potential.
What does it do?
It has several key functions. Firstly, this is where lateral roots originate. New lateral roots actually arise from dividing pericycle cells, pushing their way out through the cortex and epidermis. Secondly, in roots that undergo secondary growth, the pericycle contributes cells to the formation of the vascular cambium. And thirdly, it can also give rise to the first cork cambium in roots that develop bark. So it's a really important layer.
Lateral roots start from way inside. That's interesting. And how are the xylem and phloem arranged in this central cylinder?
The arrangement is quite distinct from stems. In most Eudicote and gymnosperm roots, the very center of the vascular cylinder is occupied by a solid core of primary xylem. This xylem core often has ridges or arms radiating outwards towards the pericycle. It might look like a star or fluted column in cross-section. Strands of primary phloem are located between these xylem ridges, alternating with them. The number of xylem ridges, or poles, varies depending on the species. We use terms like diarchs, 2 ridges, triarch, 3, tetrarch, 4, or polyarch, many.
Okay, so xylem in the middle with radiating arms, phloem in the gaps between the arms.
Exactly. And the very first xylem elements to mature, the protoxylem, are located at the outermost tips of these xylem ridges, right next to the pericycle. The later maturing metaxylem occupies the center of the ridges and the core.
Got it. Now, what about the effect of secondary growth on the root? What happens internally when a root starts to thicken, like in a woody plant?
Well, secondary growth in roots is initiated by the formation of the vascular cambium. This typically starts from patches of procambial cells that remain undifferentiated between the primary xylem arms and the primary phloem strands. Often, cells of the pericycle lying outside the protoxylem poles also start dividing and join these patches, forming a continuous, initially wavy ring of cambium.
So it connects up inside the pericycle?
Yes. This vascular cambium then begins to divide, producing secondary xylem, wood, towards the inside, and secondary phloem, bark, towards the outside, just like in a stem. As it produces more secondary xylem inwards, the cambium ring becomes circular. This secondary growth causes the root to increase significantly in diameter.
What happens to the outer layers, the epidermis and cortex?
They can't keep pace with this thickening. As the vascular cylinder expands, and typically as the first periderm, containing the cork cambium, forms, usually originating from the pericycle, the outer tissues, the entire cortex, including the endodermis with its Casparian strips, and the epidermis, are pushed outwards, cut off from nutrients, and eventually die and are sloughed off. So the mature, thickened, woody root is covered by bark, just like a woody stem.
So the original outer layers are just shed. Okay. Lastly, for roots, Let's explore root modifications. Beyond the basic, roots aren't just simple anchorage and absorption structures. They have evolved some truly amazing adaptations for different functions and environments.
They certainly have. Plants are incredibly inventive with their roots. Consider aerial roots, which grow in the air rather than the soil. Some of these, like the thick prop roots that grow down from the lower nodes of maize, corn stems, or the massive stilt roots of mangrove trees growing in coastal swamps, provide additional support and anchorage.
Like buttresses for trees.
Exactly. Other aerial roots, like the clinging roots of ivy, are modified to help the stems climb up surfaces.
And you mentioned plants in swamps. What about oxygen?
Right. In swampy, waterlogged habitats where the soil is anaerobic, lacks oxygen, some trees like mangroves or bald cypress develop specialized roots called air roots or pneumatophores. These actually grow upward out of the mud and water into the air.
Roots growing up against gravity.
Yes, they are negatively gravitropic. These pneumatophores are spongy inside with lots of air spaces, parenchyma tissue, and they have numerous lenticels on their surface. They function like snorkels, allowing oxygen to diffuse down to the submerged parts of the root system, enabling the roots to respire in the oxygen-poor environment.
Snorkel roots. Amazing. What about those orchids that grow on other plants, epiphytes?
Epiphytic orchids, which grow on tree branches rather than in soil, have highly specialized aerial roots. Their epidermis is often multi-layered, forming a spongy whitish tissue called velamen. Yes, velamen. It provides mechanical protection, reduces water loss from the root cortex, and importantly, can rapidly absorb and store rainwater or even atmospheric moisture like a sponge. In some species, the velamen even contains chloroplasts and can photosynthesize.
Photosynthetic roots.
Wow.
And for food storage, beyond just the normal parenchyma in the cortex.
Many plants have roots that are highly specialized for storage, becoming very fleshy due to an abundance of storage parenchyma packed with starch or sugars. In a carrot or a parsnip, this storage occurs mainly in the secondary xylem and secondary phloem of the taproot. The sweet potato is even more complex. It's a modified lateral root where additional secondary vascular cambia form around individual vessels, producing large amounts of storage parenchyma. And the sugar beet forms what are called supernumerary cambia, multiple concentric rings of extravascular cambia outside the original one, each producing vascular tissue and associated storage parenchyma, leading to those massive storage roots.
So they just keep adding storage layers.
Essentially, yes. It's interesting to note that the upper portion of many of these fleshy roots that we eat, like carrots or beets, actually develops from the hypocotypal, the embryonic axis below the cotyledons, not just from the true root derived from the radical. It's often a combination.
A mix of stem and root tissue.
Yes.
And we learn a lot about the fundamental mechanisms controlling root development from model organisms like Arabidopsis, right? You mentioned the positional information idea.
Yes, Arabidopsis thaliana roots have been invaluable for understanding basic principles. Its relatively simple, predictable cellular organization makes it ideal for studying cell fate determination. Those laser ablation experiments we've talked about, where specific cells are precisely removed, have clearly shown that positional control through cell-cell interaction, rather than strict adherence to cell lineage, is paramount in determining what a cell becomes. For instance, if you ablate the cortical initial cells, adjacent pericycle cells can actually switch their developmental fate and divide to replace them, becoming cortex instead of pericycle.
So cells can change their minds based on who their neighbors are or aren't.
Exactly. And specific genes are involved. For example, the TTG gene we mentioned earlier in relation to trichomes also plays a role in root hair formation. It seems to be involved in specifying whether an epidermal cell forms a root hair or remains hairless based on whether it's positioned over the boundary between two underlying cortical cells or directly over a single cortical cell. This challenges the traditional view of meristems as entirely autonomous, pre-programmed, pattern-generating machines, and really emphasizes the importance of ongoing communication and feedback between cells during development.
Fascinating level of cellular conversation happening underground. Okay, let's move above ground. Section 4. The shoot growth, leaves and flowers.
Ugh.
From the hidden world of roots below, let's now ascend to the above-ground marvels in Chapter 25. The shoot, primary structure and development. We're talking about the jutsu, which consists of the stem and its leaves. This is the part of the plant that's usually most familiar and visible to us.
The shoot is indeed structurally more complex than the root in many ways. A key difference is that the shoot is segmented. It has nodes, which are the points on the stem where leaves, and typically axillary buds, are attached, and internodes, which are the stem segments between the nodes. And unlike the root apex, which basically just grows straight ahead and produces only the root cap, the shoot apex is constantly and repetitively generating lateral organs, specifically leaves and axillary buds. These axillary buds, located in the axil, between the leaf and the stem, have the potential to develop into lateral shoots or branches.
So the stem's main jobs are support and conduction, positioning those leaves and linking them to the roots.
Primarily, yes. Stems provide physical support, lifting the leaves and positioning them favorably to capture sunlight for photosynthesis, and they serve as vital conduits, forming the connection between the leaves and roots. They transport the manufactured food, sugars, photosynthesis, downwards from the leaves via the phloem, and they transport water and dissolved minerals upwards from the roots via the xylem.
Let's zoom right into the tip, the shoot apical meristem, SM. Orchestrating above-ground growth. How is the growth tip of the shoot, the SAM, which produces all these leaves and branches and elongates the stem, actually organized? Is it like the root tip?
It has some similarities, but also distinct differences. In most flowering plants, the vegetative shoot apical meristem exhibits what's called a tunica corpus type of organization.
Tunica corpus. Sounds like clothing.
It does a bit. The tunica consists of the outermost layer, or sometimes layers, often designated L1 and L2, of cells at the very apex. These tunica cells divide primarily anticlinally, meaning the plane of cell division is perpendicular to the surface. Think of adding new bricks to the surface layer of a dome without increasing the number of layers. This contributes to surface growth.
Okay, surface layers. What's the corpus?
Beneath the tunica is the corpus, often derived from an L3 layer. Here, the cells divide in various planes, including paraclinally, meaning parallel to the surface. These paraclinal divisions add cells inwards, increasing the bulk and volume of the developing shoot apex.
So tunica for surface area, corpus for bulk. And again, like the root, cell differentiation here is mostly about position, not lineage, right?
Yes, the principle holds true here as well. Experimental evidence, like creating chimeras with genetically different cell layers, shows that the final differentiated fate of a cell derived from the SAM is largely determined by its final position within the developing shoot, overriding its original layer or lineage. Positional information is key.
Are there zones within the SAM, like the quiescent center in the root?
Yes, there are analogous functional zones. The very tip, comprising the uppermost parts of the corpus and the overlying tunica layers, acts as the central zone. Similar to the root's quiescent center, cells here divide relatively infrequently. This region is thought to maintain the pool of undifferentiated initial cells. Surrounding the central zone is the peripheral zone. This region is mitotically much more active. Cells are dividing rapidly here. This is where leaf primordia, the earliest stage of leaf development, and the beginnings of the vascular tissues are initiated. And deep to the central zone, contributing cells to the core of the stem, the pith, is the pith meristem.
Central zone like the reserves, peripheral zone for making leaves. Got it. And genetic studies, particularly in Arabidopsis, have revealed a lot about the sophisticated control mechanisms governing SAM activity and making sure it doesn't get too big or too small.
Indeed, genetics has been hugely informative here. For example, genes like shoot meristemless, STM, are absolutely essential for establishing and maintaining the SAM in the first place. Mutants lacking STM function simply don't form a proper shoot meristem. Then there's Wuschel, WS, which is necessary for maintaining the stem cell identity of the initial cells in the central zone. It's expressed in a small region called the organizing center, just below the initials, sending a signal upwards.
Okay, so WOS keeps the stem cells going. Is there anything stopping it from going crazy?
Yes. Counteracting WS are the clavata CLV genes, CLV1, CLV2, CLV3. These act to regulate the size of the meristem by oppressing WS activity. CLV3 specifically encodes a small peptide signal produced by the stem cells themselves, which then binds to the CLV1, CLV2 receptor complex in deeper layers, ultimately restricting the WS expression domain.
Wow, a feedback loop.
Exactly. The interaction between WS and CLV3 forms a crucial negative feedback loop that precisely adjusts the size of the initial cell population. If the stem cell pool gets too large, CLV3 signal increases, which dampens down WS, reducing stem cell proliferation. If it gets too small, CLV3 signal decreases, WS increases, and proliferation ramps up. It showcases really sophisticated genetic regulation maintaining meristem homeostasis.
Amazing biological thermostat. Okay, let's move down from the very tip to stem growth and primary structure. Unlike the root, you can't really divide the growing stem into neat, distinct regions of cell division, elongation, and maturation, can you? Seems more mixed up.
That's generally true. It's less clearly zoned than the root tip. This is partly because the shoot apical meristem produces leaf primordia so rapidly and close together near the apex. The inner nodes between these young leaves are initially very short. Stem length increase occurs mainly through the subsequent internodal elongation. And this elongation doesn't happen just in one zone. It can occur simultaneously across several developing internodes further down from the apex.
So multiple segments are stretching at once.
Often, yes. Although sometimes, particularly in grasses, this elongation is localized primarily in a distinct zone at the base of each internode. This zone is called an intercalary meristem. It allows grasses to regrow quickly after being grazed or mown because the meristem is at the base, not the tip.
Ah, that explains why lawns keep growing back. What about stem thickness during this primary growth?
Stem thickening during primary growth involves cell divisions occurring longitudinally along the stem axis, as well as the enlargement of those cells. In plants that will later undergo secondary growth, like most eudicots, this primary thickening is usually relatively moderate. However, in some plants that typically lack secondary growth, especially many monocots like palms or large herbaceous plants like bananas, this primary thickening can be quite substantial, sometimes massive. This occurs in a region just below the apical meristem, often called a primary thickening meristem or meristematic cap, located beneath the young leaf bases.
So palms get wide first, then tall.
Essentially, yes. They establish their mature diameter through primary growth before significant height increase. How varied is the internal primary structure of stems? We see thin stems, thick stems. There's considerable variation, but we can recognize three basic types of organization for the primary vascular tissues within the ground tissue. One, you can have a more or less continuous cylinder of vascular tissue surrounding a central pith. The vascular tissue might be truly continuous or appear as bundles very close together with only narrow interfascicular regions, ground tissue between them. You might see this in plants like basswood tilia. Two, alternatively, you can have a cylinder composed of discrete vascular bundles, clearly separated from each other by wider regions of ground tissue. This is common in many eudicots like elderberry, alfalfa, or buttercup. The ground tissue is clearly differentiated into cortex outside the bundles and pith inside. Three, the third type, characteristic of most monocots like maize, corn, or other grasses, involves scattered vascular bundles. The bundles appear almost randomly distributed throughout the ground tissue. In this arrangement, you can't really distinguish a distinct cortex and pith.
Scattered bundles in monocots. That's a key difference. And are these vascular bundles always open for potential future growth, like adding wood?
Not always.
Yeah.
In vascular bundles described as open, some of the precambial cells between the primary xylem and primary phloem remain meristematic. They don't fully differentiate. This persistent layer of precambium is the precursor to the vascular cambium, allowing for secondary growth later on. This is typical for most eudicots and gymnosperms that undergo secondary growth. However, enclosed vascular bundles, like those found in monocots or some herbaceous eudicots like buttercup, all the precambial cells within the bundle differentiate into either primary xylem or primary phloem. There's no remaining meristematic layer. Therefore, the potential for further growth, secondary growth, within that bundle is lost. Monocots generally cannot undergo secondary growth for this reason.
Open for business, secondary growth, or closed.
Got it. Now let's focus on leaves, the photosynthetic powerhouses. These are usually the principal organs for photosynthesis, capturing sunlight energy. They come in such an incredible variety of shapes and sizes, and their arrangement on the stem often seems very precise, almost mathematical.
It often is very precise. The arrangement of leaves on a stem is called phyllotaxis. There are several common patterns. Leaves can be arranged in a spiral or helical pattern, where there's typically one leaf per node, and successive leaves spiral around the stem, like an oak or ivy. They can be distichous, meaning arranged in two opposite ranks or rows along the stem, common in grasses. They can be opposite, where pairs of leaves arise at each node, typically oriented 90 degrees to the pair above and below, like in maples or mints. Or they can be whorled, where three or more leaves arise from a single node, like an oleander.
Spiral, opposite, whorled. How does the plant know how to arrange them so perfectly, often following patterns related to the Fibonacci sequence? That's been a long-standing question in botany. Several hypotheses were proposed over the years, like leaves forming in the first available space, or existing leaf primordia creating an inhibitory field, preventing new ones from forming too close, or even biophysical forces like buckling of the surface layers. However, it's now quite clear, largely thanks to molecular genetics, that the plant hormone auxin plays the primary signaling role. The currently favored auxin-based model suggests that high concentrations of auxin accumulate at specific sites on the flank of the shoot apical meristem, facilitated by auxin transport proteins like PIN1.
Auxin triggers a leaf.
Yes, these localized auxin maxima are thought to be the trigger that initiates the outgrowth of a new leaf primordium. Once initiated, this new primordium acts as a sink for auxin, depleting it from the immediately surrounding cells. This local depletion temporarily inhibits the initiation of other primordia nearby, thus creating spacing and influencing where the next auxin maximum and therefore the next leaf will form. It's a dynamic self-patterning system mediated by auxin transport.
So auxin is the traffic controller for leaf placement. Fascinating. Now let's talk about the structure of a leaf itself, the basic parts.
Most typical eudicot leaves consist of a broad, flattened blade, also called the lamina, which is the main photosynthetic surface, and usually a stalk-like petiole, which attaches the blade to the stem at a node. Some leaves also have small, often leaf-like appendages at the base of the petiole called the stipules. Leaves that lack a petiole where the blade attaches directly to the stem are called sessile leaves. This is common in many monocots, like grasses, where the base of the leaf often forms a sheath that wraps around the stem for some distance.
And we see simple leaves and compound leaves. What's the difference?
A simple leaf has a blade that is undivided, although it might be deeply lobed or toothed along the margin. A compound leaf, in contrast, has a blade that is fully divided into several distinct subunits called leaflets. These leaflets can be arranged in different ways. In a pinnately compound leaf, the leaflets are arranged along a central axis called the rachis, which is essentially an extension of the petiole, resembling a feather. Pinna means feather. Think of ash or walnut leaves. In a palmately compound leaf, the leaflets all diverge from a common point at the tip of the petiole, like fingers radiating from the palm of a hand. Think of horse chestnut or buckeye leaves.
How can you tell if something is a leaflet or a true simple leaf? Sometimes they look similar.
That's a good question. There are a couple of key distinguishing features. Firstly, buds are found in the axils of leaves, where the petiole joins the stem, but typically not in the axils of leaflets, where the leaflet stalk, if any, joins the rachis. Secondly, all the leaflets of a single compound leaf generally lie in the same plane of orientation, whereas multiple simple leaves along a stem would be oriented in different planes.
Axillary buds are the key.
Okay.
And leaf structure also profoundly reflects its environment, especially water availability, right?
Absolutely. Leaf anatomy is highly adapted to the environment. We can broadly categorize plants based on their water adaptations reflected in leaf structure. Mesophytes are plants adapted to habitats with adequate, moderate moisture conditions. Their leaves have typical features we'll discuss. Hydrophytes are adapted to living in water. If they have floating leaves, like water lilies, stomata might be present only on the upper surface exposed to air. Submerged leaves might lack stomata altogether and absorb gases directly from the water, often having very thin blades. Xerophytes, on the other hand, are adapted to arid, dry habitats. Their leaves often show features to conserve water. They might have a very thick cuticle, multiple layers of epidermis, and often numerous stomata, which might seem counterintuitive.
More stomata when you want to save water.
Yes, but these stomata are often sunken into pits or grooves on the leaf surface, sometimes filled with epidermal hairs, trichomes. This creates a layer of still, humid air around the stomata, reducing the water potential gradient and thus slowing down the rate of transpiration, water loss, when the stomata are open for CO2 uptake. It's about maximizing CO2 gain while minimizing water loss.
Clever microclimate control.
Yeah.
What about the inside of a typical mesophyte leaf? The filling.
The ground tissue of the leaf blade is called the mesophyll, meso meaning middle, esphyll meaning leaf. This tissue is highly specialized for photosynthesis. In many mesophytes, especially eudicots, the mesophyll is differentiated into two distinct layers. The upper layer, just below the upper epidermis, is the palisade parenchyma, or palisade mesophyll. It consists of one or more rows of elongated columnar cells arranged perpendicular to the leaf surface. These cells are packed tightly together and are very rich in chloroplasts, making them the primary site of photosynthesis. The lower layer is the spongy parenchyma, or spongy mesophyll. It consists of irregularly shaped cells with less densely packed chloroplasts. Crucially, there are large intercellular air spaces between these spongy mesophyll cells.
Why the air spaces?
These air spaces form a continuous network throughout the spongy mesophyll, and they are directly connected to the outside atmosphere via the stomata, which are usually more numerous on the lower epidermis. This network facilitates rapid diffusion of gases, CO2 entering from the stomata to reach the photosynthesizing cells, and oxygen produced during photosynthesis diffusing out. It maximizes the surface area of cell walls exposed to internal air for gas exchange.
So palisade layer for capturing light, spongy layer for gas exchange. Makes sense. In some plants, like corn, this distinction isn't so clear, right?
Correct. In many grasses like maize, corn, the mesophyll cells are not distinctly differentiated into palisade and spongy layers. All the mesophyll cells tend to look relatively similar.
And running through this mesophyll are the veins.
Yes, the mesophyll is permeated by vascular bundles, which we call the veins of the leaf. These veins contain both xylem for water delivery and phloem for sugar export, and are continuous with the vascular system of the stem. The pattern of venation differs between major groups. Most angiosperms, other than monocoques, exhibit netted venation, also called reticulate venation, where larger veins branch repeatedly into smaller and smaller minor veins, forming an interconnected network throughout the blade, like in a maple or lilac leaf. Most monocots, in contrast, exhibit parallel venation, where numerous veins of similar size run parallel to each other along the long axis of the leaf blade, interconnected by smaller transverse veins, like you see in a blade of grass or a corn leaf.
Netted versus parallel. And within a vein.
Within each vein, the xylem is typically located towards the upper side, adaxial side of the leaf, and the phloem is located towards the lower side, abaxial side. The small minor veins are primarily involved in collecting the photosynthates, sugars, produced in the mesophyll cells, and loading them into the phloem. The larger major veins are then mainly involved in transporting those sugars out of the leaf back towards the stem and the rest of the plant.
And these veins often have a protective sheath around them.
Yes, especially the smaller veins are typically enclosed by one or more layers of compactly arranged cells forming a bundle sheath. These bundle sheath cells often lack chloroplasts or have specialized chloroplasts depending on the photosynthetic pathway. For example, C4 plants like corn. Functionally, the bundle sheath acts somewhat like the endodermis in the root. It's thought to regulate the movement of substances between the vascular tissues and the mesophyll, ensuring that all substances entering or leaving the vein must pass through the cytoplasm of the bundle sheath cells. Some leaves also have bundle sheath extensions, columns of parenchyma or sclerenchyma cells that connect the bundle sheath to the upper and lower epidermis, possibly providing support or conducting water more directly to the epidermis.
Like little columns holding the leaf layers together. What about grass leaves specifically? They have some unique features, don't they? Like rolling up and dry.
They do. Many grass leaves possess specialized, large epidermal cells, usually located in longitudinal rows on the upper surface, called bulliform cells or motor cells. These cells are thin-walled and contain large vacuoles. It's thought that changes in the water potential, turgor pressure, within these bulliform cells cause them to swell or shrink. When they lose turgor during dry conditions, they collapse, causing the leaf blade to fold inwards or roll up along its long axis. This reduces the exposed surface area and helps conserve water by reducing transpiration. When water becomes available again, they regain turgor and the leaf unrolls.
Clever mechanical trick for water conservation.
Oh.
Okay, let's move to leaf development and abscission. How does a leaf actually form from that tiny bump on the stem, and what happens when it gets old and needs to fall off?
Leaf development starts with the initiation of a leaf primordium on the peripheral zone of the shoot apical meristem. This initiation involves specific founder cells undergoing divisions to form a small bulge called a leaf buttress. This buttress grows and develops into the recognizable leaf primordium, which elongates and typically develops marginal meristems along its edges. These marginal meristems are responsible for producing most of the cells that form the flattened leaf blade. The incredible diversity of leaf shapes we see, from simple entire margins to deeply lobed or complexly divided forms, arises from differences in the duration and location of meristematic activity and subsequent patterns of cell expansion during development.
So, leaf growth isn't indefinite like the stem can be.
Generally, no. Leaves exhibit determinate growth. Their development follows a predictable course and is of relatively short duration, resulting in a structure of characteristic, finite size and shape. This contrasts with the potentially unlimited or indeterminate growth of vegetative shoot epices or cambia.
And light intensity plays a big role in how a leaf develops too. Sun leaves versus shade leaves.
It absolutely does. Leaves show considerable developmental plasticity in response to light levels. Sun leaves, which develop under high light intensity, are generally smaller in area and thicker than shade leaves, which develop under low light conditions. This difference in thickness is mainly due to greater development of the palisade parenchyma layers in sun leaves. Sun leaves also typically have more stomata per unit area, more extensive vascular systems, more veins, and thicker epidermal cell walls and cuticles compared to shade leaves from the same plant. They are optimized for high rates of photosynthesis, but potentially higher water loss. Shade leaves are optimized for capturing scarce light efficiently.
Adapting structure to the light environment. What happens when a leaf gets old or damaged, or when seasons change? How does it fall off naturally?
That process is called leaf abscission. It's not just a passive breaking off, it's an active, genetically programmed process. It's preceded by distinct structural and chemical changes that occur in a specialized region at the base of the petiole called the abscission zone. Within this zone, two layers typically differentiate. An inner separation layer, consisting of relatively small cells with weak walls that are destined to break down, and an outer protective layer composed of cells that become heavily suberized and lignified.
Forming a seal before it breaks.
Exactly. Before separation actually occurs, the plant salvages valuable resources from the aging leaf. Reusable ions, like magnesium, potassium, phosphate, and molecules, like amino acids from broken down proteins, are transported back into the stem for storage or redistribution. This is why leaves often change color in autumn. The chlorophyll breaks down, revealing other pigments, and nutrients are withdrawn. Then enzymes are produced that specifically target and break down the pectins in the middle lamella and the cellulose in the primary walls of the cells in the separation layer. This weakens the connection. Eventually, environmental factors like wind or rain cause the leaf to detach cleanly along this weakened layer, leaving behind a leaf scar on the stem, which is already protected by that suberized protective layer.
A very neat and tidy process. Okay, before we leave the primary shoot, let's briefly touch on the vascular transition between root and shoot. We've seen that the vascular arrangement is quite different in roots. Solid core, alternating xylem, phloem, and stems, often bundles in a cylinder. How do they actually connect?
That's a great point. There has to be continuity. The change from the typical root vascular structure to the typical stem vascular structure doesn't happen abruptly. It occurs gradually within a transition region located in the axis of the embryo or young seedling, specifically within the hypocaudal root axis, the part derived from the embryonic region below the cotyledons and above the radicle. Within this region, the vascular strands essentially rearrange themselves. The xylem strands branch and rotate. The phloem strands shift position to achieve the stem pattern while maintaining continuous connections for water and food transport throughout the plant's life. It's a complex anatomical transition ensuring seamless integration.
Smooth plumbing connection. Now, the ultimate expression of shoot development for many plants is the flower. Let's explore flower development, the reproductive apex. How does a vegetative shoot tip turn into a flower?
The switch from vegetative growth to reproductive development, leading to the formation of a flower or an inflorescence, a cluster of flowers, marks a profound change at the shoot apex. It usually signals the end of the indeterminate growth of that particular apex. The vegetative apex transforms into a reproductive apex, or floral meristem flourescence meristem. This transformation involves significant physiological changes triggered by hormones and environmental cues like day length, and distinct structural changes. The apex often becomes broader and more dome-like compared to the vegetative SAM. Importantly, the development of a single flower is a determinate growth pattern. It produces a fixed number of floral organs and then stops growing.
And the floral parts, sepals, petals, stamens, carpels, they develop in a specific order, don't they?
Yes, typically they arise in sequence from the floral apex, usually starting from the outside and moving inwards. Sepals first, then petals, then stamens, the male reproductive parts, and finally carpels, the female reproductive parts, in the center. This sequence is thought to reflect their evolutionary origin as modified leaves.
And genetics has given us huge insights here, too, with the ABC model.
Absolutely. Studies of homeotic mutations in flowers, particularly in Arabidopsis and snapdragon mutations, where one type of floral organ is replaced by another, like petals developing where sepals should be, led to the formulation of the elegant ABC model of flower development. This model proposed that three classes of genes, A, B, and C, act in overlapping concentric regions or whorls of the developing flower to specify the identity of the floral organs. For example, A genes alone specify sepals. A plus B genes together specify petals. B plus C genes specify stamens. And C genes alone specify carpels. The genes essentially act like a combinatorial code.
So different combinations of active genes determine which part forms where.
Exactly. The model has since been expanded to the ABCDE model to include D-class genes involved in ovule development within the carpels, and E-class genes, also called sepallata genes, which are necessary for the activity of A, B, and C genes, and seem to play a role in specifying overall floral organ identity. It shows that even something as complex and beautiful as a flower is assembled under precise genetic control using a relatively simple combinatorial logic.
It's like a genetic instruction manual for building a flower.
You could think of it that way. And specific genes trigger the whole process. For instance, a gene called AEY-LFY in Arabidopsis encodes a key transcription factor that essentially acts as a master switch, assigning floral fate to meristems that would otherwise have remained vegetative or produced leafy shoots. Plants with the leaky mutations simply cannot make flowers, they just keep making leaves.
Amazing control. Finally, for the shoot, let's look at stem and leaf modifications. Beyond the norm. Plants get incredibly creative with their stems and leaves, modifying them way beyond the typical forms for specialized functions like climbing, defense, or storage.
They really do. Evolution has produced some fantastic modifications. For support, tendrils are common. These are slender, coiling structures that help plants climb. Some tendrils are modified stems, like those of grapes or Boston ivy, which forms adhesive discs at the tips. But most tendrils are actually modified leaves or parts of leaves, like the terminal leaflets of a pea's compound leaf that become coiling tendrils.
Climbing adaptations. What about defense? Spikes and thorns.
Yes, sharp structures for deterring herbivores are common modifications. It's useful to distinguish between them botanically. Thorns are modified stems that arise in the axils of leaves, where a branch would normally form, and are often hard and pointed, like in hawthorn or citrus trees. Spines are modified leaves or parts of leaves, like stipules. The spines of cacti are a classic example. The entire leaf is reduced to a sharp spine, and photosynthesis is carried out by the fleshy stem. Barberry spines are also modified leaves. Prickles, like those on a rose stem or blackberry cane, are technically different. They are sharp outgrowths originating from the cortex and epidermis of the stem, not modified stems or leaves. They can be snapped off relatively easily.
Thorns are stems, spines are leaves, prickles are skin outgrowths. Good distinction. And for food storage, beyond the fleshy roots we discussed, stems and leaves get in on the act too.
Absolutely. Underground storage organs derived from shoots are very common. Tubers, like the familiar white potato, are the swollen tips of underground stems, specifically rhizomes or stolons. They are packed with storage parenchyma filled with starch. The eyes of a potato are actually the nodes on this modified stem, each containing axillary buds that can sprout into new shoots. Bulbs, such as an onion or tulip, are technically large buds. They consist of a small, conical, underground stem with numerous fleshy, thickened leaves attached to it. These modified leaves are the primary storage organs, packed with food reserves. Corms, like those of a gladiolus or crocus, look similar to bulbs externally but are structurally different inside. A corm consists primarily of thickened, fleshy stem tissue, which is the main storage organ. The leaves attached to it are usually thin, papery, and protective, not fleshy storage leaves like in a bulb.
Tuber is stem, bulb is leaves, corm is stem. Got it.
Right. And even things we might not think of as modified, like the familiar head of cabbage, is essentially a very short vegetative stem bearing numerous thick, overlapping leaves that store food. These are all ingenious ways plants have evolved to store energy and nutrients, often underground, to survive unfavorable periods and fuel future growth.
Plants are just incredibly adaptable architects. Okay, let's move to our final section. Section 5, secondary growth thickening and strengthening. So far, we've focused almost entirely on primary growth, how the plant extends in length from its apical meristems and forms its primary tissues. But many plants, especially the woody ones like trees and shrubs, also grow substantially in girth, becoming thicker and stronger year after year. This process is secondary growth, and it's the focus of our final chapter from this section, chapter 26, secondary growth in stems, though it also occurs in roots, as we touched on.
Secondary growth is indeed a remarkable process. It allows plants to increase their diameter, provide stronger mechanical support, and produce new conducting tissues over long periods, enabling them to reach great sizes and live for many years, sometimes even centuries or millennia. This thickening growth involves the activity of two specialized lateral meristems, which run parallel to the long axis of stems and roots, unlike the apical meristems at the tips. These are the vascular cambium, which produces secondary vascular tissues, wood and inner bark, and the cork cambium, which produces the outer bark, or periderm. Secondary growth is characteristic of all gymnosperms and most angiosperms, with the significant exception of monocots, which generally lack the necessary lateral meristems.
Okay, two lateral meristems driving growth and girth. Before we dive into the mechanics of those, let's quickly define plant life cycles, annuals, biennials, and perennials, as this classification often correlates strongly with whether a plant undergoes significant secondary growth.
That's a good point, as life cycle strategy influences growth patterns. Annuals are plants that complete their entire life cycle, from seed germination to producing their own seeds, all within a single growing season, and then they die. Think of marigolds, zinnias, or many crop plants like wheat or corn. They typically have only primary growth. Biennials require two growing seasons to complete their life cycle. Usually, they undergo vegetative growth, producing roots, stems, leaves in the first season, often storing food reserves. Then, after overwintering, they bolt in the second season, producing flowers and seeds, and then die. Carrots, parsley, and foxgloves are examples. They might have limited secondary growth, especially in the root. Perennials are plants that live for many years. They can be herbaceous perennials, where the above-ground parts die back each winter, but the roots or underground stems survive to produce new shoots the following spring, like peonies or hostas. Or they can be woody perennials, trees, and shrubs, where the above-ground stems persist year after year and typically exhibit significant secondary growth. Secondary growth is primarily found in these woody perennials.
Annuals, biennials, perennials. Clear distinction. Now let's get into the engine of secondary growth, the vascular cambium, generating wood and inner bark. This is the meristem responsible for making the stem or root thicker with vascular tissue. Where exactly is it located?
The vascular cambium is typically a thin cylindrical layer of meristematic cells located between the primary xylem and primary phloem in stems and roots that are capable of secondary growth. In stems with discrete vascular bundles, it arises from the residual procambium within the bundles, vesicular cambium, and often also from parenchyma cells between the bundles, intervesicular cambium, linking up to form a continuous ring.
So it forms a complete cylinder. What kinds of cells does it contain?
The vascular cambium itself contains two distinct types of initial cells. One, fusiform initials. These are vertically oriented, elongated, spindle-shaped cells. They are much longer than they are wide. When they divide periclinally, parallel to the surface, they produce derivatives that differentiate into the cells of the secondary xylem, wood, towards the inside, and the cells of the secondary phloem, inner bark, towards the outside. These form the axial system of the wood and bark. 2. Ray initials. These are horizontally oriented, shorter, more cuboidal or isodiametric cells. They are arranged in short radial files. When they divide, they produce derivatives that form the vascular rays. These are radial sheets or ribbons of parenchyma cells that extend horizontally through the secondary xylem and secondary phloem, forming the radial system.
Fusiform initials for vertical elements, ray initials for horizontal rays. What do the rays do?
The vascular rays are crucial for lateral transport of water and minerals across the wood, for storage of reserves like starch and lipids within the wood and bark, and for transport of sugars radially from the phloem into the deeper tissues.
Okay, so the vascular cambium is constantly adding new layers. Wood, secondary xylem on the inside, inner bark, secondary phloem on the outside.
Exactly. Throughout the growing season, the cambial initials divide repeatedly. For every division producing a xylem derivative, which happens more frequently, it also produces a phloem derivative outwards. And occasionally it divides anticlinally, radially, to increase the circumference of the cambium itself as the stem or root girth increases. In temperate regions, this cambial activity is seasonal. The cambium is typically dormant during the winter and becomes reactivated in the spring, stimulated by hormones, particularly auxin, produced by the developing buds and young leaves. This flush of new growth in spring is why bark often slips or peels easily off the wood. At that time, the newly formed, thin-walled cambial cells and xylem derivatives are easily broken.
Ah, that's why. Okay, so that's the vascular cambium making wood and inner bark. What about the outer protection? That's the job of the cork cambium, forming the outer bark.
Yes, as the vascular cambium adds layers inwards, increasing the girth, the original epidermis and cortex are stretched, eventually rupture, and are sloughed off. A new protective layer needs to form further out. This is the role of the cork cambium, or phellogen. The cork cambium is the second lateral meristem. It usually originates from cells in the cortex, just beneath the original epidermis, or sometimes from deeper tissues like the phloem or even the pericycle in roots. Like the vascular cambium, it's typically a cylindrical layer.
And what does it produce?
The cork cambium divides periclinally to produce tissues both outwards and inwards. To the outside, it produces cork cells, also called phellem. To the inside, it produces phelloderm cells. Together, these three layers, the cork cambium phellogen itself, the cork phellem it produces outwards, and the phelloderm it produces inwards, collectively constitute the periderm. The periderm is the functional replacement for the epidermis in stems and roots undergoing secondary growth. It becomes the outermost layer of the bark.
What are those cork cells like? You mentioned suberin before.
Yes, cork cells are key to the periderm's protective function. As they mature, their walls become heavily impregnated with suberin, that waxy, waterproof substance. They often deposit lignin as well. This makes the cork layer highly impermeable to water and gases. Importantly, cork cells are typically dead at maturity, their function residing in their persistent, suberized walls. They form a tough protective barrier against dehydration, physical damage, and pathogens.
Dead, waterproof, protective cells. What about the philoderm on the inside?
The phyloderm cells produced towards the inside by the cork cambium are usually living cells. They typically resemble the parenchyma cells of the cortex from which the cork cambium originated. There's usually only one or a few layers of phyloderm compared to potentially many layers of cork. They may function in storage.
Okay, so periderm cork plus cork cambium plus philoderm, and you mentioned lenticels earlier, those spots for gas exchange. They're part of the periderm system.
Yes, lenticels are essential features of the periderm. Since the cork layer is largely impermeable, the living tissues inside the stem or root still need a way to exchange gases, like oxygen for respiration, with the atmosphere. Lenticels are localized regions within the periderm where the cork cambium produces cells that are more loosely arranged, often rounded, with large intercellular air spaces instead of tightly packed suberized cork cells. These porous regions allow for vital gas exchange through the otherwise impermeable bark. They often appear as distinct raised or horizontal streaks on the bark surface and typically form beneath the original stomata present in the epidermis.
Breathing pores in the bark. Okay, let's clarify the term bark. All tissues outside the vascular cambium. What does that encompass exactly?
It's a common term, but botanically, bark is defined quite broadly. It includes all tissues located external to the vascular cambium, so it's everything from the vascular cambium outwards. We can usually divide the bark into two regions. One, the inner bark. This consists of the living, functional secondary phloem, which is actively transporting sugars. Two, the outer bark. This includes all the tissues outside the innermost cork cambium. It typically consists of any non-functional, older secondary phloem that has been pushed outwards, plus all the periderm layers, cork cambium, phyloderm, that have formed. If multiple peridomes have formed sequentially deeper within the stem, the outer bark will include all the dead tissues isolated between them.
Why non-conducting phloem in the outer bark? I thought phloem transported sugars.
It does, but only the most recently formed secondary phloem, the part right next to the vascular cambium, the inner bark, is actually functional in transport. Sieve elements, the conducting cells of the phloem, are relatively short-lived. In many trees, they function for only one growing season before their sieve plates get blocked and they die or become non-functional. So, as the vascular cambium produces new secondary phloem each year, the older phloem from previous years gets pushed outwards, crushed, and becomes part of the non-conducting outer bark. Only the innermost layer of phloem remains active in transport.
Ah, okay. So only the fresh stuff works. And this layering of periderms leads to different appearances of bark on different trees, right?
Yes, absolutely. The appearance of the outer bark depends largely on how subsequent cork cambia arise. If new cork cambia form as discontinuous, overlapping patches deeper in the secondary phloem, they will isolate patches of older tissues, leading to a bark that fissures and flakes off in scales or plates. This is called scale bark, common in trees like pine, oak, or pear. If new cork cambia form as continuous concentric cylinders deeper in the phloem, the older bark layers will not be able to expand in circumference and will eventually split vertically, but the bark might appear smoother or peel in rings. This is called ring bark, seen in trees like birch or cherry or vines like grape. Commercial cork harvesting from the cork oak relies on the tree producing exceptionally thick layers of cork, vellum, from its persistent cork cambium.
Scale bark versus ring bark. Interesting textures. Finally, we come to the substance itself. Wood, the secondary xylem. Wood is such a defining feature of trees, and structurally, it's essentially just accumulated secondary xylem.
That's exactly right. Botanically speaking, wood is secondary xylem, the tissue produced inwards by the vascular cambium. It makes up the vast bulk of the stem and root in woody plants. Commercially and structurally, wood is often broadly classified into softwoods and hardwoods. These terms can be a bit misleading about actual density, but generally. Softwoods are the woods produced by conifers, gymnosperms like pine, fir, spruce. Conifer wood is typically structurally simpler. Its axial system consists mainly of tracheids, which serve for both conduction and support, and parenchyma cells. Vessels are absent, except in a small group called the natales. Conifer wood often contains resin ducts, tubes lined with secretory cells producing resin. Hardwoods are the woods produced by woody angiosperms, flowering plants like oak, maple, birch, balsa. Angiosperm wood is generally more complex and specialized. It contains vessel elements, usually the primary water conductors, Tracheids, various types of specialized fibers, which are the main support cells, and axial and ray parenchyma cells. The presence of vessels and abundant fibers often makes hardwoods structurally more diverse and often denser than softwoods, though balsa is a very light hardwood.
Softwood, conifers, mostly tracheids, hardwood, angiosperms, vessels, fibers, etc.
Got it. And those famous growth rings or annual rings we see in tree stumps or furniture, how do they form?
Growth rings are a direct result of the seasonal activity of the vascular cambium, especially in temperate regions with distinct growing seasons and dormant periods. Each visible annual ring typically represents the secondary xylem produced during a single growing season, usually 1 year. The ring structure arises because the wood formed early in the growing season, early wood or spring wood, looks different from the wood formed later in the season, late wood or summer wood.
How are they different? Early wood is formed when growth conditions are optimal. Plenty of water, active shoot growth. It typically consists of wider diameter vessel elements in hardwoods, or tracheids in softwoods, with relatively thinner walls. Its primary function is efficient water conduction. Late wood is formed later in the season when growth slows down. It typically has narrower diameter cells with much thicker walls and is denser. Its primary function shifts more towards providing structural support. This visible contrast between the less dense, lighter-colored early wood of 1 year and the denser, darker-colored late wood of the previous year creates the distinct boundary that we see as an annual ring.
Wider cells early, narrower cells late. Makes a visible line. So counting rings can tell us a tree's age.
It gives a very good estimate in many cases, yes. Dendrochronology, the science of dating tree rings, relies on this. However, it's not always perfectly straightforward. Sometimes abrupt environmental changes during a single growing season, like a drought followed by rain, can cause the tree to form a false annual ring, making it look like 2 years' growth occurred in one. And in tropical regions where growth might be more continuous throughout the year, distinct annual rings may not form at all.
So usually reliable, but with caveats. What about the difference we often see between the lighter colored outer wood and the darker colored central wood in an old log or stump?
Ah, yes. That's the distinction between sapwood and heartwood. The lighter colored, physiologically active outer portion of the wood is the sapwood. This is the wood that is actively conducting water and minerals, the sap, up from the roots. It also contains living parenchyma cells in the rays and axial parenchyma, which store carbohydrates and help defend against pathogens. As the tree ages and the trunk increases in diameter, the older inner layers of sapwood gradually cease to function in water transport. They undergo a transformation process to become heartwood.
What happens during this transformation? Why does it get darker?
Several things happen. The living parenchyma cells in the wood die. The trachary elements, vessels and tracheids, often become blocked. And crucially, the dying parenchyma cells often synthesize and deposit various secondary metabolites, things like oils, gums, resins, tannins, and phenolic compounds into the surrounding cell walls. These substances often darken the wood, increase its density, make it much more resistant to decay by fungi and insects, and can give it distinctive colors and odors. It's thought that forming heartwood might be a way for the plant to safely sequester potentially inhibitory or toxic metabolic byproducts away from its living active tissues, essentially turning the core of the tree into a durable, non-functional, chemically protected pillar.
So sapwood is living and conducting. Heartwood is dead, stored waste, and decay resistant.
That's a good summary. And sometimes, especially as sapwood transitions to heartwood or in response to wounding or pathogens, the vessels can become physically blocked by tyloses.
Tyloses, what are those?
Tyloses are balloon-like outgrowths. They actually come from adjacent living ray or axial parenchyma cells, which protrude through the pit openings in the vessel walls and inflate into the vessel lumen, the central cavity. They can partially or completely block the vessel. This can help prevent the spread of air embolisms, air bubbles, or fungal pathogens through the xylem network. They are often abundant in heartwood.
Like little plugs forming inside the pipes. And one last thing about wood structure, those knots we see in lumber.
Yes, knots are simply the bases of old branches that have become embedded within the main trunk as the vascular cambium of the trunk added successive layers of secondary xylem around them year after year. A tight knot was from a living branch when it was overgrown. A loose knot was from a dead branch. They represent the history of the tree's branching pattern recorded in the wood.
The tree's history written in wood. Okay, that covers secondary growth. Let's wrap up. What an absolutely incredible journey we've taken through the angiosperm plant body. It's really something. From that first asymmetrical division of a single zygote laying down the body plan, all the way to the complex structure and towering trunk of a woody tree. It's a story of precision, adaptation, and continuous, highly regulated growth.
It really is. We've seen how fundamental patterns like apical basal and radial polarity guide early embryogenesis, how those primary meristems, the protoderm, procambium, and ground meristem, lay the foundational blueprint for the entire plant. And then how these differentiate into the specialized dermal, ground, and vascular tissues that build up the functional organs, the roots anchoring and absorbing, the stems supporting and conducting, and the leaves capturing the energy of the sun.
And the ingenious adaptations just kept coming. From the Casparian strip acting as that selective gatekeeper in the roots, forcing everything through cellular checkpoints, to the tunicle corpus organization managing growth at the shoot tip, to leaves fine-tuning their internal structure for sun or shade, and stems and leaves modifying themselves into specialized structures like tendrils for climbing, spines for defense, or massive storage organs like tubers and bulbs for survival. Even that simple cactus spine being a modified leaf just highlights the incredible versatility built into the system.
And then finally, that remarkable process of secondary growth, how the vascular cambium and cork cambium, those lateral meristems, continuously add new layers of wood and bark. This allows plants to increase dramatically in girth, providing robust support and protection, enabling them to truly stand the test of time, all while constantly maintaining that delicate balance between growth, transport, and defense.
So, what does this all mean for you listening in? Hopefully, the next time you look at a plant, whether it's a tiny seedling bravely pushing its way through the soil, or a delicate leaf unfurling towards the sun, or the massive, weathered trunk of an ancient tree, you'll remember this intricate dance. Remember the journey from a single cell, the precise organization of tissues, the sophisticated genetic controls dictating cell fate based on position, and the absolutely ingenious adaptations that have shaped its form and function. It really gives you a new appreciation, doesn't it?
It truly makes you appreciate the profound complexity, the resilience, and maybe even the sheer biological artistry of plant life that's unfolding all around us every single day. It's easy to take them for granted, but there's so much going on inside.
Thank you so much for joining us on this deep dive into the fascinating world of angiosperm structure and development, guided by the insights from Raven Biology of Plants. We hope you've gained a new perspective on the green world that sustains us.
Until next time, keep exploring, keep questioning, and definitely keep an eye out for the amazing biology unfolding in the plants all around you.